Every living organism, from the smallest bacterium to the largest whale, is composed of cells. These microscopic compartments house the machinery of life, carrying out the essential functions that define living organisms. Understanding cells is crucial to understanding life itself. There are two primary types of cells, prokaryotic and eukaryotic. Prokaryotic cells are simpler and smaller, representing the earliest forms of life on Earth. In contrast, eukaryotic cells are larger, more complex, and house a variety of internal compartments called organelles. These differences in structure reflect differences in function and evolutionary history. Both prokaryotic and eukaryotic cells share some common features, such as a cell membrane and genetic material. However, the differences between these cell types have profound implications for the diversity of life. Prokaryotic cells are the oldest and simplest form of life on Earth. They are characterized by their small size and lack of a true nucleus. In prokaryotic cells, the genetic material is located in a region called the nucleoid, where a single circular chromosome resides not enclosed within a membrane-bound nucleus. In addition to lacking a nucleus, prokaryotic cells also lack other membrane-bound organelles found in eukaryotic cells such as mitochondria and chloroplasts. Despite their simplicity, prokaryotic cells are incredibly diverse and occupy a vast range of habitats. There are two main types of prokaryotic cells, bacteria and archaea. Bacteria are the most familiar type of prokaryotes and are found almost everywhere, in soil, water, air, and even inside other organisms. Examples of bacteria include Escherichia coli, which is found in the intestines. Streptococcus pneumoniae, which can cause pneumonia, is a type of pathogenic bacterium commonly found in the respiratory tract, and cyanobacteria, photosynthetic bacteria, often called blue-green algae. Another group of prokaryotic cells is the archaea. Many archaea are extremophiles, meaning they thrive in extreme and often surprising environments. Extremophiles can be classified as thermophiles, halophiles, acidophiles, methanogen, alkaliphiles, and psychrophiles. Thermophiles thrive in temperatures between 45 degrees Celsius and 80 degrees Celsius, while hyperthermophiles prefer above 80 degrees Celsius, even up to 105 degrees Celsius in deep-sea hydrothermal vents. Halophiles thrive in high-salt environments like salt lakes, salt mines, or even salted foods. They grow best in 15–30% salt. That's like eating a meal with 3 to 6 tablespoons of salt, which would be dangerously salty for humans. Methanogens are a type of archaea that produce methane gas. They live in places without oxygen, like swamps, wetlands, and the stomachs of cows, where they break down waste and release methane, a gas that can also be used as a source of energy. Eukaryotic cells, in contrast to their prokaryotic counterparts, are larger and significantly more complex. The defining feature of eukaryotic cells is the presence of membrane-bound organelles, including a nucleus that houses the DNA. The genetic material is organized into multiple linear chromosomes in eukaryotic cells, unlike the single circular chromosome found in prokaryotes. The cytoplasm of eukaryotic cells is also highly compartmentalized, containing a variety of membrane-bound organelles, each with specialized functions. For instance, mitochondria are responsible for energy production through cellular respiration, while chloroplasts in plant cells carry out photosynthesis. Examples of eukaryotic cells include those found in plants, animals, fungi, and protists. The evolution of eukaryotic cells marked a significant leap in complexity, paving the way for the incredible diversity of life we see today. The differences between prokaryotic and eukaryotic cells are fundamental and have significant implications for understanding the evolution and diversity of life. One of the most obvious differences is size. Prokaryotic cells are much smaller than eukaryotic cells, typically 10 to 100 times smaller in diameter. This difference in size is related to the presence of membrane-bound organelles in eukaryotic cells. This compartmentalization, or the presence of organelles, allows for greater efficiency and specialization of cellular processes. Here's another key difference between prokaryotes and eukaryotes that's super important: how they divide. Prokaryotic cells use a process called binary fission, which is fast, simple, and super efficient. Basically, the DNA gets copied, the cell grows a little, and then, snap, it splits into two identical cells. No fuss, no fancy machinery needed. The process begins with the replication of the cell's single circular DNA molecule. Once the DNA has been copied, the cell elongates and the two identical DNA molecules move to opposite ends of the cell. Next, a dividing wall called a septum starts forming in the center. Finally, the cell splits into two genetically identical daughter cells. Eukaryotic cells have a more sophisticated approach to division, and they use two different strategies depending on the purpose. Mitosis is the process behind how the body grows, heals, and replaces old or damaged cells. In mitosis, one parent cell divides into two genetically identical daughter cells, each with the same number of chromosomes as the original. Meiosis is used to create gametes like sperm and eggs. This process involves two rounds of division, resulting in four genetically unique cells, each with half the number of chromosomes. So, while prokaryotes divide in a matter of minutes, eukaryotic cell division can take hours. But it's super precise and tightly regulated. Prokaryotes represent an older lineage of life, while eukaryotes are thought to have evolved from prokaryotic ancestors through a process of endosymbiosis. This understanding of the differences between prokaryotic and eukaryotic cells is essential for numerous applications, including medicine, agriculture, and biotechnology.